Good morning, or maybe good afternoon or good evening, depending on where you're checking in. My name is Edward Nelson. I'm a sales, service, and applications engineer here at Nanosurf. In today's webinar, I will be describing a method to implement automated nanomechanical analysis of blended polymers. But first, maybe a little bit of background. Mechanical analysis is the study of the material properties of a sample. This can include things like the adhesion, stiffness or elastic modulus of a sample, or indentation. Now, the nano part of nanomechanical analysis refers to the probe diameter. We're going to be using probes for the most part that are less than 100 nanometers in diameter. This means the resolution is less than 100 nanometers or in the nanoscale. What kind of applications is nanomechanical analysis valid for? Well, a lot of times it's used with cultured cells or tissues. So for example, in lab-on-chip applications, the cell growth and mobility of cells are affected by the substrate stiffness. For tissue screening, for example, for cancer screening of tissue cells, that depends on the stiffness of the cells in the tissue. For hydrogels and surface coatings, so for things like catheters or stents, the stiffness or the adhesion of that material is really important for being effective in its application. And for this talk today, I'm going to be talking about polymers and polymer blends. So how the stiffness of the polymer in a mixture changes based on mixing parameters, etc. So I want to back up a little bit and talk about what is exactly an AFM-based nanoindenter. It differs a little bit from a standard indenter that you may be used to. Now, the basic concept is we have a calibrated force sensor, and with an AFM, we call this a cantilever. At the end of the cantilever is a sharp probe with, again, diameters less than 100 nanometers typically. We use a laser which reflects off the end of the cantilever and hits a position-sensitive detector. When there is no deflection at all, or if the cantilever is no force on the cantilever, we get no deflection. Now, when we push into a surface, the cantilever will bend. This will cause the laser to deflect, and we can now measure the deflection of the cantilever, which is proportional to the force. So we can use how much we've pushed in, the vertical displacement of the cantilever, and the force, and use that relationship to determine the mechanical properties of the surface. So let's take a look at how this looks from a signals point of view. So here I have the axis of a graph. On the x-axis, I have the tip sample separation. So this is the distance above the sample or into the sample that we are. On the y-axis, we have cantilever deflection. And remember, this is the force on the cantilever. As we start far away from the surface, we approach the surface. At one point, we start to interact with the surface, so we make contact. And then we push further into the surface, the force on the cantilever increases. Now, we can just take a look at baseline deflection. This is zero force. We can calculate the maximum force on the sample. This is what we set beforehand as the set point. We also know where we are on the surface, where the surface is, and how far into the material we indent, so the max indentation. In the in contact region, so this region here, we can calculate the slope, and from that, we can calculate the elastic modulus. Now, there's a little bit more complicated than that, and I'll get into the different models we can use right now. On the retract, so if we pull backwards, we get a lot of cases some adhesion with the sample. So the cantilever will stick to the sample, and there'll be a rupture force where we return back to the baseline zero. This maximum value to the minimum here in this course, which is the maximum adhesion, can also give us some more information about our material properties. So I mentioned that the slope in contact is related to the elasticity, and there are a number of contact mechanics models, and these are just 5 models, examples here. The first three are models in which there is no adhesion between the tip and the surface, and they describe kind of three different types of probes. The first is kind of the most famous or the most common model. It's called the Hertzian contact mechanics model. This is you use this when you want to model a spherical tip, so a bead or a ball at the end of a AFM cantilever with a large radius and again no adhesion. We also have models that describe maybe a pyramid indenting into a surface. This is common. This is similar to what you have with an actual AFM cantilever pyramid, and a cone interacting with a surface. Now the equations on the right describe essentially the shape of the probe. So we have the force on the probe or on the surface is equal to or is proportional to some value here, which is E. This is the reduced elastic modulus. This takes into account the Poisson ratio of the surface, a term that describes the radius, in this case of the sphere, and D is the indentation depth. We also have similar equations or other equations describing pyramid and a cone. Now, if you take into account adhesion, we have two other models to describe that. One is the DMT model. This is typically used for stiffer samples with a small tip radius and a small adhesion energy gamma. We also have the JKR model, which is mostly used for softer samples with a large tip radius and large adhesion energy. And now the equations are a little bit more complicated than the first three, but if we take a look, we see that the force is proportional again to this first term, which is exactly the same as the Hertzian model. So in contact, we are using the Hertzian model. And then we have the second term, which describes the adhesion. For the JKR model, it's again even more complicated, but we have this initial term, which is the in contact region, and then this second term, which describes the adhesion, and we have this extra term A, which is the contact area between the tip and the surface. Now, both of these DMT and JKR models, if we have no energy, adhesion energy, excuse me, so if gamma equals 0, so this second term disappears and this second term disappears, and we return back to the Hertzian equation. So these two equations can be used with also no adhesion energy. We just revert back to Hertz. So I've been saying automated nanomechanical analysis. So and we call that ANA, A-N-A. What is ANA? Well, ANA is a complete hardware and software solution for real-time automated data collection and analysis. All right, so for today's agenda, in this webinar, I will show you how we can use ANA to acquire the material properties of two different mixed polymer samples. The first sample is a two-component polypropylene with ethylene propylene rubber domains. The second sample has four components, two different thermoplastics, polypropylene, and also some rubber domains. Now these samples were prepared with a cryomicrotome, so they have a flat surface. All the measurements I'm going to show here were all done with a Flex ANA system. Now, at this moment, I should acknowledge that the contributions from Dr. Dalia Yablon from Surface Care for preparing the samples and for the discussions of these results. Now, you might be asking, why is automation important? Well, to get accurate results, we need a lot of statistics, or we need a lot of data to get accurate statistics. Either high density data within a single area to account for surface heterogeneity, or even better, multiple areas across a surface to account for local differences in maybe sample preparation or annealing or any other local variation. At the same time, for some samples, it is not possible to take the measurements quickly, and I'm referring to viscoelastic samples. For these reasons, automation saves time in the long run. So I mentioned viscoelastic materials. These can be things like wood or tissue or some sort of polymers, some types of polymers. And what happens when you measure these materials is that if you measure the sample slowly, you get one type of force curve. But if you measure quickly, the slope of the force curve changes, meaning the contact mechanics change. So depending if we measure slowly or quickly, the results of that sample change. So in order to get realistic results, we really have to go slowly to measure viscoelastic samples to get the static elastic modulus. So what this means is that automation will save us time. We need large data sets. We can't necessarily take this data very quickly, especially if we're working with viscoelastic samples. So the automation really makes this a lot easier. And let me give this as an example. Here on the right, we see 8 different samples. On each sample, I want to measure 20 areas to get enough statistics. At the same time, within each area, I'm going to measure 16,000 measurements, take 16,000 measurements within that. In all, that represents almost 2.6 million different measurements are going to be taken. So if I was doing this manually, so going to each one of these points, doing a force map, manually moving the sample, approaching, taking another force map, etc., it would take me about 120 hours to complete this measurement. Now, if I was working 8 hours a day, that represents 15 working days, so about 3 weeks, 3 working weeks to take this measurement. On the other hand, with automation, I can do this entire measurement in about 80 hours. The reason is the software, the ANA software, automatically goes to each point, measures, analyzes the results, and goes to the next point, etc. Not only is it faster to take all the data, but since it's running 24 hours a day, that can be done in about 3 full days of work, so 5 times faster. Now, what is in the ANA hardware? To take the actual measurements, we have the AFM scan head. It's an XYZ tip scanner. It allows you to take maps that are up to 100 by 100 microns in size and 10 microns in Z range and height. This is suitable for flatter samples. So this could be cryotomed polymers, spin coatings, thin films, things like that. Cells, a confluent layer of cells. The second part of the hardware is an overview camera. This allows you to see every single sample within your measurement space. And to tie it all together, we have a motorized stage with a 35 by 35 millimeter XY range. This moves the sample to underneath the force sensor in the AFM scan head where you can take the measurement. The last bit, which is completely optional, but we can also provide a 100 micron Z range actuator for soft samples or particularly sticky samples. So things like tissue, which are very rough, or some biological samples which are very sticky or high adhesion. You might need that extra 100 micron Z range in order to measure those samples. Now, the 3 main components, the scanner, the overview camera, and the motorized stage, they all need to talk to one another. And that's where the ANA software comes in. ANA software coordinates the overview camera with the motorized stage and with the AFM scan head. So everything works together nicely. Before we can take a measurement, we need to make sure the hardware and the software are working together nicely. The first thing we need to do is coordinate the stage position and the camera, the overview camera. ANA will do this automatically using image recognition algorithms. The second thing we need to do is coordinate where the AFM probe is and where the samples are on the stage. Now, this is more of a manual process, but what we can do is on a sample, we can select an area that we can see on the overview camera, and ANA will do the rest after that. Now, because at the end, we want our results to be quantitative, we're going to need to have a calibrated cantilever, a calibrated force sensor. And we'll calibrate this force sensor in 2 parts. The first is we need to find the relationship between the detector signal and deflection. This is what we call deflection sensitivity. Now, if you recall this diagram from before, if I push against a surface, the cantilever will bend. Now, the laser that's reflecting off the end of the cantilever, it comes against this position-sensitive detector and registers a change in position or a change in deflection, and this is a signal that we register in volts. But what we really want is what's the deflection in meters, not in volts. So how do we get about finding this relationship? Well, to do that, what we use is instead of a soft surface here, we use a hard, what we would call an infinitely hard surface. It can be something like mica or sapphire or something like that. And as we approach the surface and make contact with the surface, we get a force curve where the change in z position, delta z, relative to the change in signal here, delta v, will give us the relationship we need. So if we take the ratio of delta z over delta v, this is the inverse slope, we get the deflection sensitivity of the cantilever. So for most cantilevers, most values, expect values between 50 and 500 nanometers per volt. The second calibration we have to do is of the relationship between the actual deflection and the force of the cantilever, right? We want to measure actually force at the end. So to do this, we have to remember that the AFM cantilever is a spring. So meaning the force is proportional to deflection. And if you recall back in physics, Hooke's law says that this is a scalar equation, that the force here is proportional to the displacement x by a constant k. So k is our spring constant. So how do we measure the spring constant of an AFM cantilever? Well, in the ANA software, we will experimentally measure it. We will actually measure it using what is called the Sater method. And to do the Sater method, we need actually a couple of things that we can to apply the Sater method. The first is we need to know the dimensions of the cantilever, both the width and the length. And this we can just ask the manufacturer of the cantilever what they are. We can measure them optically, etc. We also need the quality or Q factor of the cantilever in air. This is how good of a resonator we have. And finally, we need the resonance frequency of the cantilever. These last two, the Q factor and the resonance frequency, this we can measure with the ANA software. And if you're curious about different methods of calibrating AFM cantilevers, there's a nice reference by Nancy Burnham in Nanotechnology. And of course, the Sater method, John Sater wrote about it in the Review of Scientific Instruments. Now, let's talk about the first calibration, deflection sensitivity. In ANA software, this is a single button calibration. We placed a hard sample underneath the cantilever. You can see the cantilever here. You see the reflection of the cantilever below it on this piece of sapphire, or of silicon, excuse me. We press the start button, the software does everything else. The second is the spring constant. Again, single button press. We calculate the thermal fluctuations of the cantilever. This is the power spectral density. So what we see here is the first resonance of the cantilever. From here we can decide, or we can measure the resonance frequency, which will be at this peak, and then the width of this will give us the Q factor. Those are displayed here under resonance frequency and Q factor. Again, the width and the length we know from the manufacturer, and from there we can get the spring constant. So let's talk about the ANA workflow. After we've calibrated everything, we've done hardware calibration. So we calibrated the stage with the cameras, with the scanner. We've calibrated the force sensor. So now we know, based on how much signal we get in volts, we know how much force is being applied. The last thing we need to do is the actual measurement and analysis. So here is our samples. This is the cryotome sample. I have two samples here. The one on the left is a 2-component sample. The one on the right is the 4-component sample. In ANA software, what we get is an overview camera. And from here, we can assign 3 areas to measure on the first sample, and 3 areas to measure there. These are denoted by these squares there. We can change the size of the actual area of the measurement point. So this is 10 by 10 microns, it says here. And we can change the resolution, so 128 by 128 number of points and lines. This would be about 16,000 data points per sample. The next thing is that we want to set up the actual individual force curves. So some important parameters is how much applied force do we apply to a sample, so 20 nanonewtons in this case. How fast do we take the measurements? This is 2 microns per second. I believe for this sample actually went around 10 microns a second. And this parameter here tells me how far should I withdraw the cantilever between going to different areas in the sample. So if I have a very rough sample, or if I have a height difference between the 2 samples, I can withdraw further away from the sample before approaching again. Now, the last thing to set up is while the ANA takes the actual measurements, it can also give us some real-time analysis of each measurement as it comes in. And this is where we would set that up. So here under Data Analysis tab, I first can set up the model I'm going to use to model the results. And I have access to the 5 models I described before. In this case, I want to use the DMT model because I have a relatively stiff sample, I have a small tip radius, and I have adhesion. Here is where I would set up the rest of that model, the individual components, so the tip radius and the Poisson ratio. And I also have some tilt correction and noise suppression. Then is the actual acquisition, and this is what the screen would look like during an actual acquisition. I have the actual image here, the overview image. I have indicated where I'm taking the current force curve. While the force curves are being acquired, I get the actual individual force curve as it's being measured, so I can look at it, make sure it looks good. It's what I expect. I also have aggregate histograms of the adhesion, modulus, stiffness, or contact point for both the forward and backwards that I can build here. I have two locations here. I have here a real-time map being shown of the aggregate 2D maps of also those 4 channels: adhesion, modulus, stiffness, and contact point, and I can select either forward or backwards. At the left, I have on the bottom the actual results for each individual force curve as they come in, or above that, I have the aggregate values, the average values of all of them. Now, because this is, we can see that this is actually a complex material here. This is the 4-component material. The average values might not mean really as much as maybe the individual values. So let's take a look at the results of this measurement of the DMT model on this 2-component sample. This is why I chose the DMT model to analyze it. And as we can see in the 2-component, there are certainly 2 areas that we can identify here. There's maybe the first component with a lower elastic modulus, and this other brightly colored area with a kind of higher elastic modulus, or above 1 or around 1 gigapascal. If we look at the actual force curves, we can see the difference between the force curve in the softer areas, the black areas. It has higher adhesion, and the slope is not as steep as the second region here, which has a lower adhesion and a pretty stiff force curve, and it's a much steeper force curve there. Now, because we're working with automation, this can lead to having very big data sets with millions of data points which we have to analyze. The ANA software will analyze the force curves automatically, but when we have to actually analyze the results of those force maps, we might have to refer to an expert to process these. Now, machine learning can help process these big data sets, and it does so programmatically. So for this analysis, I'm going to use what is called the Gaussian mixture model of clustering. This is an unsupervised learning technique, meaning the computer or the machine will look for patterns in a data set without any predefined labels. The Gaussian mixture model, this is a probabilistic model and assumes that all the data points are generated from a mixture of a finite number of Gaussian distributions with unknown parameters, meaning every single distribution has a mean and a variance. We're going to use GMM to categorize each force curve. The benefits are that it's fast, but it requires a lot of training data. Fortunately, we have a lot of data because we've automated it. But you also have to know the number of components. So this might not be suitable for all samples, but in this case, I know I have a 2-component and a 4-component sample. So this is the reason I've decided to use this clustering model. So let's take a look at the first 2-component sample. Again, this is the raw data. If I apply the Gaussian mixture model to it, it correctly identifies the 2 components. Component 1, which is here highlighted in blue, it has a mean stiffness of 0.11 gigapascals, or about 100 megapascals. The other component is this gray color here with a mean of 0.84 gigapascals. If we take a look at the histogram now, this is how the machine learning clustered the 2 samples. We can see that the component 1 takes up about 16.5% of the total area of the sample, and component 2 is the rest. But what we see is that here in the middle, the machine has to decide where one component is versus the other. Now, what I think we'll see is that around the edges of these components where there's mixing involved, we're going to get something in between here. So what we can do is introduce a mixing component. So this is going to account for this this area between these 2 Gaussians here. And what I've done is I've set a mixing component of 100 megapascals around this area here. And now what we see is that, in fact, yes, it's this area highlighted in red here is actually accounting for this mixing between the 2 components. Now, if we take that out of the total area here, we find that the actual component 1 has a mean of closer to 80 megapascals. Remember, from 110 megapascals, it went down to 80 megapascals. Component 2 is now 0.85, so that increased a little bit. And now we can account for the mixing in between. And now we can see also what percentage of the surface is one component versus the other. So component 1 is 14.3%, component 2, 81.3%. So that's how we can use machine learning to help analyze the results of an ANA experiment. Now, this is a very simple example. There's only 2 components. What happens when we apply it to a more complicated component or more complicated sample like the 4-component sample? So let's take a look at the results of the ANA software from ANA. Using the DMT model, we see that in this 15 by 15 micron scan, there are 128 data points per line. We can easily see there are clearly 4 different components here. There's a larger, there's a background darker component in the background. Here's one, here's another one here, and then this lighter kind of wavy or snake-like pattern almost everywhere. That's the 4th component. In the histogram, it's also clear. We have 1, 2, 3, 4 different components here. Now, this also highlights the benefits of nanomechanical analysis or testing. The rubber domains within the sample are between 100 and 500 nanometers in width. So, for example, this right here is 350 nanometers wide. If we're using a traditional nanoindenter or a bead, like a Hertzian bead, to analyze these, some beads are 5, 10, 20 microns. We won't be able to get all this nice detail using a traditional indenter. But going back to using machine learning, if we take the raw data and now pass it through our same model or Gaussian mixture model, we see now that the 4 components are accurately identified. We have in the background, which was darker here, this blue component with a 0.15 gigapascal stiffness. Here's the second component with 0.41 gigapascals. Then this, I guess, snake-like or wavy pattern, 0.71, and then the stiffer pattern here, 1.3 gigapascal. We can see that the second component takes up about almost a third of the total area here, with maybe the remainder 2/3 taken up almost evenly by the other 3. Now, in this case, I also noticed that there is going to be some mixing. So if you look at the histograms and how it decides where one component is and where the other one is, I'm sure there's going to be some mixture here at 0.25 gigapascal, and here also. And what that's going to correspond to, and we can see that in the image here, that this second component is labeled throughout the center of this. And so what I see is maybe a little bit of an error in this Gaussian mixture model, where it's assigning this mixture between the blue region and the first component and the third component, and it's assigning it to the second component. And the reason is that because it's a mixture, it's going to have the properties of either of these. So this could benefit actually from adding this mixture coefficient to this sample and try to see if we can actually separate this out a little bit. So, in summary, nanomechanical analysis is a technique to get mechanical information with nanoscale resolution. Large data sets and long measurement times require automation to simplify the workflow. ANA automates the entire nanomechanical workflow, from calibration to the actual measurement and analysis. In this webinar, I showed you an ANA measurement, two different multi-component polymers, and I showed you how machine learning algorithms can further assist with the analysis. Thank you for your attention, and I really hope you enjoyed this webinar, and I'll be happy to take any of your questions.